Well, first I want to thank the organizers for the invitation, the opportunity to sort of share your research with the communities and groups that have most empowered it is humbling and really an honor. So I was asked to talk about sort of a translational approach to childhood disorders, and we accomplished that mostly using genomics. And so what I thought I would start with is giving you an overview of our process, and then maybe speak specifically about one of the specific disorders. So as a physician scientist, I usually start these things by thinking about a particular disease or a clinical phenotype, and then thinking about the epidemiology and the demography of that to be able to inform an appropriate genetic model. So you might end up with very rare diseases that fit the Mendelian mode, for which there are very few genes and very high effects, or you could be at the other end of the spectrum. And so once we've sort of thought about a genetic model to use, then we sort of recruit cohorts accordingly, and then we can apply sort of the modern genomic technologies. And that really forms a nidus for discovery. And that's the starting point to then funnel our way through sort of mechanism and using models to understand how we could best sort of have a targeted intervention, with the hope that ultimately we might be able to close the loop in going from bedside to bench and back. So we're doing this on a number of different fronts. You can see the diseases that we're interested in here, and all of these have a very heightened prevalence in populations of African ancestry, which makes this more prescient. So what I thought I would do today is to talk you through some of the work we've done on childhood malnutrition using this kind of genomics approach. So severe acute childhood malnutrition is still a major global health issue, with a very high prevalence worldwide, and particularly on the continent. And the major problem, of course, is that it's associated with a very high mortality, both indirectly and directly, particularly in children under the age of 5. Now, you can say, well, you know, the way that you treat severe malnutrition is sort of just have food security. But we've known that for a while, and still there are a number of sort of geopolitical and climate issues that often intervene to keep that from being a reality and make it still a very prescient disorder to consider. So what's less well considered sometimes with severe malnutrition is that there are kind of two prevalent forms. The first is the sort of edematous form, which goes by the names of kwashiorkor or marasmic kwashiorkor, and is characterized by sort of multi-system involvement, including the hair, the skin, and then pretty much every other organ system you could imagine. And is characterized by swelling or edema of the body. And this form is classically much more difficult to treat and has a higher mortality when compared to the non-edematous form, which is also known as marasmus, which is predominantly characterized by wasting. And although we've known these two forms for some time, it's actually not 100% clear why you get one and not the other. And some of the proof of principle of that is that if you look at the treatments, it's the same for both forms, despite them having different outcomes and different pathophysiologies. And this is not a new disease. So there are those who have sort of pointed to references to the Israelites wandering around in the desert and comments about their feet not becoming swollen as some of the earliest references to kwashiorkor. But the first official mention in the medical literature was in 1933 from Cicely Williams, who was a Jamaican pediatrician who was working in Ghana, in Accra at the time. And since that original description, there have been a number of studies, and here this is just the publications in PubMed that list kwashiorkor. And those studies have predominantly focused on biochemical, physiological, and nutritional differences between the two. And so there's this very large catalog of differences that you can observe between the two groups in their physiology. But despite all these studies, it's still actually not known that there's any single clear definitive etiology. But what is known is that there's no consistent difference in the diet or infectious exposure. And so that has shifted a lot of the focus back to more innate issues, which includes genetics. And so we have taken on thinking about edematous malnutrition or kwashiorkor as a complex multifactorial disorder. And so one of the physiologic metabolism defects that's been observed, that's sort of coming to the fore as maybe most relevant to the disorder, is one, carbon metabolism, specifically the conversion from methionine to homocysteine and back, which is responsible for 80 to 90% of the methyl groups that are produced, including the methylation of DNA, which you see here. And stable isotope studies that were done in Jamaica many years ago demonstrated that when acutely ill, the children who have the edematous form have a much slower turnover in this cycle than those in the non-edematous form. And when recovered, however, there's no difference between the two groups. So we thought that this was a good place to start in terms of thinking about our genomics approach. And that's because DNA methylation is mitotically heritable. By that I mean that when the DNA is replicated, the new strand has to have the same methylation marks placed on it. And so you could imagine that in cells that have a high mitotic rate and they're turning over at some rate, that you could find that with each sort of generation of cell proliferation, this sort of limiting availability of methyl groups and substrates might mean that you could actually eventually see differences in DNA methylation in a bulk way between the two groups, and that might have an insight into the underlying pathophysiology. And so we decided to study this using two cohorts from Jamaica and Malawi. And these were all children, all aged between sort of 0 and 3 years, who had buccal cells done. And those buccal cells were taken in and around the time of their original diagnosis. So this is a acute group, we refer to them as DC. And the numbers are actually relatively evenly split between Jamaica and Malawi. We're also fortunate to have a group of adults from Jamaica who had survived childhood malnutrition and are now adults, and they're healthy otherwise. And so all these individuals were typed on the Illumina 450 array or the EPIC array. And so we're looking at just under 500,000 CpG methylation sites across the genome. And so the first thing we did was we just asked, do we see evidence of differences in methylation? So of those sites where we could see, strongly, significantly differential methylation, almost all of those sites at a Bonferroni corrected significance level, almost all of those sites are hypomethylated in edematous relative to non-edematous, which are the red dots. We also saw that this effect was both similar in magnitude and direction in Jamaica and Malawi, and that the CpG loci that were involved are spread all throughout the genome, which you see represented here on the outer ring, and the black dots represent the individual loci on the inner ring. So to reduce some of the false positives that we sometimes get with sort of a single locus kind of approach, we clustered these associated regions into differentially methylated clusters between the groups, and you can see those here in the red dots in the outer ring. But interestingly, when we looked at the adults and compared their methylation status, we see no difference between edematous and non-edematous. So this is strongly in keeping with what the stable isotope biochemical studies had shown us, which is that these primary differences are occurring in that acute nutritional event. And then when they get better, there's no longer those differences. So we wanted to give this some additional context. So we looked at where these differentially methylated sites were. And you can see that relative to background, we see an enrichment over the gene body. So the relationship between sort of DNA methylation and gene expression is well understood at promoter regions to be sort of inversely correlated. And so we had some RNA and DNA paired samples from some of the children. So you can see at the promoter region here, there's an inverse correlation, which represented the y-axis, such that you would expect low methylation, high gene expression, and vice versa. But at the gene body, we see both evidence of some negative correlation, but also some positive correlation. And so the overall picture is that although it's hypomethylation, you're seeing this very complex transcriptional response, which is likely to be influencing the complex physiology that we see. So we thought about, well, what genes might be involved in that? And so we put these in the context of gene ontology terms, and you can see again that we're getting some biological relevance to the underlying biology. So responses to exogenous things like nutrition, things that affect sort of water and solute movement related maybe to edema, and then things that relate to sort of anthropometry and body size related to nutrition. And in fact, if we kind of looked specifically at the individual genes, we could see lots of examples of ones that have been brought out in the literature of severe malnutrition, including sort of fatty liver in sort of rat models, of malnutrition, genes that have been implicated in changes in glycosaminoglycan content in the small bowel of children with kwashiorkor. And then we also saw a number of genes that are related to immunity. And in particular, have been shown to be disrupted in sort of overnutrition states, so obesity or elevated BMI with DNA methylation. And in fact, if we took all of the genes that we observed and sort of went to the GWAS catalog and just asked what, if any, genes are associated with these particular, what disease traits are represented by these genes, we can see again that we see this preponderance of sort of cardiovascular and metabolic disease and things to do with body mass and anthropometry, where you see an accumulation of these kind of genes occurring. So that said, well, maybe we're on the right track biologically, and allowed us to develop this kind of molecular framework as a working framework for going forward. And so that would say, well, in the face of this stress, we have this aberrant 1-carbon metabolism, and then there are downstream disruptions in sort of DNA methylation and other related processes. And that then gives rise to the two phenotypic differences that we see between edematous and non-edematous malnutrition. And the nice thing about this framework is that it does allow for things like the microbiome. So studies have shown where you have changes in the gut microbiome, you get a shift in towards gut bugs that have a very different 1-carbon metabolism. And would also be consistent with a recent mouse study where they fed the mice a sort of diet that's very similar to the one that you see in Malawi and has been associated with malnutrition, where after about 2 weeks, you can see this accumulation of fat, which is these red globules here in this liver biopsy. Whereas the same mice given the same diet, but with choline, which is a methyl donor, you actually don't see that accumulation of fat. Now, choline has a lot of other different uses, so this is not exactly like a slam dunk, but it is highly suggestive that we're on the right track. So then we wanted to kind of give this a little bit more sort of shape. And so as a human geneticist, I was really interested in how genetic variation might influence this. And so you could think about the first way, which is that it might influence the DNA methylation as these sort of so-called methylation quantitative trait loci, or QTLs. And actually, we did find evidence of this. So you see that illustrated here, where the degree of methylation in the edematous group is dependent upon the underlying genotype in the acutely ill state, but you don't see that same effect in the non-edematous acute or in the completely recovered group. And this kind of context-dependent DNA methylation QTLs, we found at about a quarter of the sites where we had differential methylation. And so we kind of view these as maybe sort of fine-tuners of the hypomethylation. That is, the kind of things that might make the difference between where you're more severe or less severe, or why you might have more fatty liver or skin or hair changes in one case than the other. But there are other ways that your genetic variation might play a role. And so you could think about, well, what about the genes that are related to directly related to 1-carbon metabolism? And so we did look at this using our full cohort of 700 individuals from Jamaica and Malawi, all genotyped on the H3 Africa array. And then we just looked for any locus that has been associated with 1-carbon metabolism in the literature. And then we sort of focused initially on the SNP genotypes that were related to these 105 genes. And so, if we look at those in a Manhattan plot, so again, each dot is a SNP, the height reflects how significant it is. And at a gene level cutoff, and certainly in keeping with the QQ plot and its deviation from from expected. We can see there are at least 3 loci that meet this kind of threshold. And of course, like then the question is, well, what are these 3 loci? So, all of these loci are going to be related to 1-carbon metabolism in some way. And so PRCL2 is the first one. The exact role of PRCL2 is not precisely known, but as a locus, it's certainly been associated with folate levels and replicated as such. GABBR2 is actually a GABA receptor, but it's the same idea that sort of the upstream region as a locus has been associated with homocysteine levels fairly consistently. And then we have SHMT1, which is actually the enzyme that converts 5-methyl tetrahydrofolate into tetrahydrofolate in the remethylation of methionine. And so at least the top SNPs are sort of all culminating on this area where we're sort of remethylating methionine, which of course is an essential amino acid. So if we look at that in a bit more detail, what we can see is that at GABBR2 and SHMT1, we see there's a higher frequency of the top alleles in the edematous in the non-edematous versus edematous. So this is more of like a protective effect, right? So protecting from edematous forms. And actually the variant in SHMT1 is a strong EQTL, particularly in the thyroid, which is a relevant tissue to malnutrition. And the direction that you see here would be consistent with the sort of resulting enzyme changes that would fit our original hypotheses. At PRCL2, we can see that all of the sort of top SNPs there are all within this block of LD. And it has the same kind of features where it's a sort of a protective type of effect from edematous malnutrition. So we kind of wanted to take our top SNPs and also put them in a different, broader context. And so one thing we thought is that, if you are going to survive severe malnutrition, that might be the kind of thing that would have a fairly significant selective effect, much the way that sort of like surviving childhood malaria might. And so we started to look at signatures of selection in our data. And so what we're plotting here is a measure of sort of more distant selection on the y-axis and more recent selection on the x-axis. And you can see that in the tails of this, there's certainly a number of SNPs related to 1-carbon metabolism on this side. And in fact, a couple of these SNPs are actually the same SNPs that have been significantly associated in PRCL2. So that's sort of a positive thing. But interestingly, this is what we saw in Malawi. But in Jamaica, although the sort of overall distribution is the same, we don't see the same SNPs sort of coming out in the same way. I'm going to come back to that. So then we thought, well, that's the top SNPs. So what about 1-carbon metabolism SNPs in general when we sort of consider it, given that we do have the other 2 million and some SNPs that are on the H3 Africa array? And so to sort of evaluate this in more detail, we sort of said, well, we're going to create a hypergeometric distribution by, say, starting with how many SNPs would one expect to see having a p-value less than 0.001. And so we have that number from the data set, and we then permute the case control status and do sort of 10,000 permutations of that so that we can develop a hypergeometric distribution. And then we can just ask for actually our observed data set. What is the where does that fall? And that's where this red line is, showing that 1-carbon metabolism SNPs, the number of 1-carbon metabolism SNPs that are significantly associated at this cutoff is well out in the tails of that distribution. So you might say, well, of course, because you're sort of looking in a particular pathway and just comparing it against sort of randomness. But if we do that with a different pathway set of SNPs, so here's sphingomyelin, we've done it with ketones as well, we don't see the same sort of broad distribution. And if we use one of these other pathways as our background and do the permutation there, again, 1-carbon metabolism SNPs are still way out in the tails. And in fact, if we change the p-value thresholds, instead of 0.001, we sort of go all the way down to a more permissive cutoff of 0.01, we see that the resulting z-score still has 1-carbon metabolism SNPs way out in the tails of this distribution. So that was sort of encouraging that, this overall approach might be interesting. But what we did notice is that we still saw these differences between Malawi and Jamaica. And of course, We also saw this when we looked at the haplotypes that are associated in each of our top regions. So even in a region where there's very strong and similar linkages to equilibrium, the top haplotype that's associated is basically the mirror in one group versus the other group. And where we have less linkages to equilibrium, then it's even more of a mess. So we're not at anywhere close to a causal allele in any way. And what we're likely dealing with here is differences related to their genetic ancestry. So, so in one sense, we're fortunate in that we can see that all of these on-principle components, they all sort of cluster together as African samples, but you see a very tight cluster of the Malawi samples in red, whereas the Jamaican samples are much more diversely spread over these first three principal components. And that sort of spoke to us to maybe think more about admixture in these individuals. So again, here, each column is a different individual, and the different colors represent sort of relative parental ancestries. So what you can see is that, looking at the ancestry of Jamaica, it's most closely related to the Yoruba, the West African in Nigeria, which is not unsurprising. And then there's some European ancestry as well, similar to the Seth from Utah. Whereas for Malawi, we see an ancestry that looks a lot more like Central Africa and southern parts of Africa as well. And so, when you think about the prevalence of kwashiorkor and edematous disease, which is much more common in sort of central and southern parts and gets less common the further north you come, you could imagine that there might be sort of a specific ancestry component that's carrying a susceptibility allele. And it might then, say, for instance, like this pink region here, might have different frequencies as you move, and that might explain some of the differences that we're seeing between Malawi and Jamaica. And so one way that we're trying to get around this is to think more about local ancestry. This is just an example from one individual from Jamaica, where you can sort of find this distribution. You can refine your association studies by knowing what the ancestry is at a particular locus and adjusting that way. And we hope that that will give us a sort of increased power to be able to tease apart these observations. So of course, this doesn't really necessarily answer the question of why you get one and not the other, although it likely contributes to it. And so we are, of course, interested in looking at a larger GWAS, for which we think we're probably going to need some larger numbers. And so, if anybody's interested in collaborating on that, we'd be very interested to hear from them. But I'm going to otherwise just stop there, just to sort of emphasize that there is this reemerging role for one carbon metabolism, which we see somewhat bolstered by the genomics that we're doing. And that this integration of different omics can give you different and unique insights into some of these disorders. And then, as I said, we're working on this. We're sort of trying to push forward on all fronts that we can. And of course, as I mentioned, are very interested in sort of expanding the footprint here with willing collaborators. So I just want to end by acknowledging the many, many, many people who are involved in bringing this kind of work to fruition, and in making a shameless plug that in our new position at the NIH, we have postdoctoral positions available for anyone who's inclined towards sort of population genetics and genetic epidemiology. Feel free to contact me. And then with that, I am happy to take questions.